So first of all, when we talk about the sperm transport, we know that once the mating occurs, the semen from male animal is deposited in the female genitalia, and depending upon the species, the site of deposition of semen varies. So, like in case of cattle and sheep, the semen volume is small and is ejaculated into the cranial end of the vagina as well as the posterior end of the this cervix. While in case of horse and swine, as the volume of the ejaculate is more, so it is deposited through the relaxed cervical canal into the uterus. So this is the table showing site of ejaculation in different species. So in case of rabbits, it is in the vagina. In case of monkeys, cattle, sheep, in the vagina only. But the semen has a high concentration of sperms in it. In case of cattle and sheep, and the site is uterus in case of horse, dog, pig, as well as rodents. So when we talk about the sperm transport in the female reproductive tract, the transport is of three types majorly. One is rapid transport, then colonization of sperm reservoir, and slow release and transport. So we will discuss these three one by one, how it happens. So when we talk about rapid transport, it happens immediately after AI or mating. When the animals mate, the semen is deposited inside the female genitalia, and these spermatozoa, they penetrate the cervical mucus layer, which is at the time of estrus is quite liquid-like and allows the penetration of spermatozoa through it. And some spermatozoa are very quickly transported through this cervical canal. This phase may take 2 to 10 minutes and is facilitated by sperm motility. Obviously, the sperms gain motility once they enter the female genitalia, as well as the contractile activity of the myometrium and mesocephalus. So, when the animals are in their courtship and the coitus occurs, the the myometrium of the uterus, it gains a contractile activity which is actually meant for facilitation of this sperm transportation. So some of the spermatozoa, they reach internal os of the cervix immediately or within 1 to 3 minutes of AI or mating. So when fertilization occurs, only a critical number of spermatozoa reach the site of fertilization, rest all are filtered out. So another thing that happens to these sperms is when they enter the cervix, there are mucosal folds which form cervical crypts. So these spermatozoa get trapped into those crypt-like structures, which are nothing but जैसे आप मान लो कोई गड्ढा type हो in the mucosa of the cervix. So these spermatozoa, they get trapped into those crypts and they form reservoirs. From these reservoirs, the sperms later on, they get released slowly. The more number of spermatozoa enter the cervical reservoir or the cervical these crypts, there are more chances of fertilization because there will be more number of spermatozoa available for this slow transport as well. So in the species where ejaculation occurs in uterus instead of this cervix, the sperm reservoir, they are localized into uterotubal part or in the endometrial glands, like in case of pig and dogs. And in other species where the ejaculation occurs in the vagina or the cervix, the sperm reservoir will be in the cervix. Another kind of transport is slow release and transport. So after this, there is numerous number of sperm reserves available in the female genital tract. The spermatozoa, they kept on releasing from these reservoirs for a prolonged period, slowly, over a long period of time. So this massive number of sperms, they reach the site of fertilization, which is actually prevented by various anatomical as well as physiological barriers, which include presence of these fluids in the uterus or the cervical mucus, or you can say the this balance of hormones. So how this sperm transport occurs in the uterus? Majorly, यूट्रस में जो स्पर्म ट्रांसपोर्ट होता है, that is aided by myometrial contractions, which help the transport of these sperms into and through the uterus towards the oviducts. So the presence of sperms in the uterus, it induces endometrial leukocytic response as well, which helps in phagocytosis of excess sperms, or you can say defected or dead sperms. क्योंकि नॉर्मली क्या होता है, ल्यूकोसिटिक रिस्पॉन्स इज एक्टिवेटेड इन द बॉडी जनरली जब भी कोई भी फॉरेन बॉडी बॉडी में एंटर करती है, तो ल्यूकोसिटिक रिस्पॉन्स गेट्स एक्टिवेटेड. So these sperms are foreign to the female body. So introduction of these sperms into the uterus, they induce endometrial leukocytic response as they are foreign bodies to the female genitalia. So this leukocytic response, it leads to phagocytosis of the excess sperms. So this way also the very excessive number of sperms get filtered, and only a few and productive, or you can say very fertile kind of sperms, they get transported further into the oviduct for fertilization. So once these spermatozoa cross the uterus, they are transported into the oviduct. So in the oviduct, the transport of these sperms is very unique. So what happens in the oviduct is, at one end of the oviduct, from the base of the uterine horn or this oviduct, the sperms have to be transported through it towards the ovum, and at the other end, when the ovulation occurs and it is taken up by the ovum is taken up by the infundibulum, so it will be transported towards the uterus. So there are two kinds of opposite directions movement. One is conveying the spermatozoa towards the ovum, and another is conveying the or transporting the ovum from the infundibulum towards the uterus. So this pattern and rate of sperm transport through the oviduct, it is controlled by several mechanisms, which includes this peristaltic and antiperistaltic movement of the oviduct, and there is contraction of oviductal mucosal folds as well, and the mesosalpinx has a fluid inside, so it also produces currents and counter currents, which is created by the ciliary action of the cells of oviductal mucosa. So the frequency and amplitude of these contractions of oviduct, it is controlled by ovarian hormones and is also controlled by PGF2 alpha, which controls the contractile movements of the oviductal musculature. So there is one endocrine control of sperm transport as well. So what happens is, there is endocrine these hormones present in the female genitalia. So the penetrability of spermatozoa, it is inhibited if there is progestational cervical mucus. Means during luteal phase, this cervical mucus becomes thick, which is not favorable for this penetrability of the spermatozoa. So by this mechanism, the female genitalia is protected against any unnecessary exposure to the foreign proteins of the semen. So there is role of oxytocin, sympathetic and parasympathetic nervous system also. Obviously, with the nervous system, with the activation of this nervous system, the various movements and the various production, they are controlled. So which controls the sperm transport. There is a role of epinephrine, acetylcholine, and histamine as well, but it is very, it is not that extensive. So once these spermatozoa are transported through the uterus towards the oviduct and reach near the ovum, so the process of fertilization occurs. So what this fertilization is, it is a multi-step phenomena which is initiated by interaction and subsequent fusion of male and female gamete, which are sperms and the ovum, and result into the formation of a zygote, which is actually single cell form of a new individual. From this single cell, a complete new individual or complete new embryo, or later on which is called as fetus, will be produced. So this fertilization process occurs in a very systematic manner. So later during this lecture, I'll show you one video as well, where you will be very much clear about what this fertilization process is all about and how it happens. It is an animated kind of video. So the various events that occur during the fertilization process are, first of all, the spermatozoa, they encounter and penetrate the cumulus oophorus, which is the external most loose layer of the ovum, as we read during the past 2 3 lectures. And as you must have read about this female and this male reproductive system and the sperm structure also. So there is activation of acrosome of the sperm and inner acrosomal membrane. It contracts. It contacts. Sorry, there is spelling mistake. It contacts the zona pellucida of the ovum, where enzymes exposed on the membrane surface, they allow penetration of this sperm into the perivitelline space. If you remember the this structure of the ovum, you will understand all these terms: zona pellucida, perivitelline space, everything. Okay. So what happens is, the equatorial region of the sperm, it attaches and fuses with the vitelline membrane, which stimulates the completion of the second meiotic division. Means, before once when this ovulation occurs, there was completion of first meiotic division, which led to the release of first polar body. So once this sperm attaches with the vitelline membrane and stimulates the completion of second meiotic division, means there will be release of second polar body in the perivitelline space, and the ovum will, the other part of the nucleus will remain inside the cytoplasm of the ovum. So there will be one large male pronucleus and there will be one small female pronucleus, which form following the extrusion of this second polar body into the perivitelline space. So these pronuclei are nothing but the initial stage of the further the nucleus which will be formed further. So these male pronucleus and female pronucleus, they come close and they fuse together. They have nuclear envelopes outside them. So once they come close, this nuclear envelopes, it disperses, and the two pronuclei, they are combined, and the prophase of the first mitosis division, it starts. So this is the pictorial this depiction of how this fertilization occurs. So this is a sperm which approaches the, this is oocyte or ovum. Once this sperm approaches the this ovum, it comes in contact with it. Acrosomal reaction, see, this acrosomal reaction occurs, which leads to release of hydrolytic enzymes from the acrosome of the sperm. And once this hydrolytic enzymes are released, they allow the penetration of the sperm through this to reach this perivitelline space, This is the perivitelline space. So the sperm gets entry through this perivitelline space and it further enters. But once it comes in contact with this zona pellucida, there are these cortical granules present just next to the zona pellucida. So once this sperm head comes in contact with the vitelline membrane, these cortical granules, they get activated and they release the proteinases and other components which form a plug, thick plug like structure which is visible over here. And this perivitelline space fluid, it becomes thickened, which does not allow this sperm body to enter inside the ovum, and just this sperm nucleus gains the entry inside, and the sperm nucleus gets transported towards the female pronuclei, and the two pronuclei, they combine further. So these cortical granules, they fuse with egg plasma membrane and render the vitelline layer impenetrable to sperm. So once just a one when just even a one sperm cell comes in contact with this vitelline membrane, there is formation of this impenetrable plug around whole of the zona pellucida, which prevents entry of further or more number of sperms into the ovum. So this process may ensures that there is just one sperm that can enter the ovum. Otherwise, there are millions of sperms present around. So they will be getting entry into the ovum, and there won't be formation of any zygote. Rather, there will be some chromosomal aberrations. So this is nature's this you can say nature's way of protecting this ovum from millions of sperms, rather just to make sure that just one sperm enters the ovum. So this is another picture showing how this fertilization occurs. So this is a sperm cell which is approaching towards the ovum. This is activation and release of hydrolytic enzymes from the acrosome of the sperm, and with the help of these hydrolytic enzymes, the sperm head gains entry into the perivitelline space. So once this sperm head comes in contact with this zona pellucida, there is there is activation of cortical granules, and the this this plasma of the ovum and the plasma of the sperm, they fuse together, which leads to this entry of male pronuclei into the into the egg, and this is egg nucleus, this is sperm
मैम आपकी
nucleus.
आवाज बहुत कट रही
आवाज कट रही है अभी ठीक है
मैम कट जाती है बीच बीच में
एक्चुअली ये जो इंटरनेट ये है ना डेटा, दिस इज क्वाइट अनस्टेबल, तो वो बीच बीच में उसकी जैसे वेरिएशन आती है तो वो कटता है उसकी वजह से. सो एम आई ऑडिबल नाउ?
यस मैम यू आर ऑडिबल.
Okay. So this egg nucleus as well as sperm nucleus, they come in close contact and they fuse together, which forms zygote's nucleus. So this is another picture depicting this entry of sperm nucleus into the ovum nucleus. So during this fertilization process, there are three critical events. One is sperm migration between the cumulus cells. Another is sperm attachment and migration through zona pellucida. And the third one is fusion of sperm and ovum plasma membranes, which lead to entry of this male pronuclei into the into the ovum, and then fusion of sperm and ovum pronuclei. So There are evidences that a substance produced by cumulus oophorus of rabbit ova, it stimulates sperm motility, right? So this factor also plays a role in sperm ovum encounter, and with these peristaltic contractions in the ampulla, the chances of ovum and sperm contact, they are increased. Similarly, there is one hyaluronidase enzyme present in the bull acrosome and aryl sulfate in the boar acrosome. These also help in this dispersion of cumulus oophorus cells, because there is one layer called cumulus oophorus cells. So the sperm has to penetrate through those cells so as to reach the ovum. And also when the spermatozoon encounters and penetrates into the cumulus oophorus, there is cellular disintegration of this cumulus oophorus cells, which uh, leads to gain of entry of this sperm into the ovum. So also there is first polar body already present in the perivitelline space, and with this fertilization process, the secondary oocyte is already present in the cytoplasm, but this polar body will be in perivitelline space. Once this polar body is formed, it will be pushed out of the this zona pellucida and will be in perivitelline space only. So also the inner acrosomal membrane, it contacts the zona pellucida, where enzymes are exposed on the membrane surface of the zona pellucida, which allow the penetration into the perivitelline space. This is just a repetition of the points which I have already told you. So the equatorial region of the sperm head, it attaches as well as fuses with the vitelline membrane, which stimulates the completion of second meiotic division, which will lead to the formation of the second polar body in the ovum, which will be released into perivitelline space only. So the large male pronucleus and the small female pronucleus, they come close, which is followed by extrusion of the second polar body into the perivitelline space. So these polar body, they migrate to the oocyte center, where the nucleus envelops these disperse and, sorry, this is not polar body, this is actually pronuclei. Sorry, this is a mistake. This pronuclei, they migrate to the oocyte center, where the nuclear membrane or envelope disperses and the prophase of first mitosis, it begins. So right immediately after fertilization, the ovum surface changes. Like I told you that this cortical granules, they release and they solidify this perivitelline space so as to prevent the fusion of additional spermatozoa. If this mechanism fails and there is no hardening, or this you can say hardening of the perivitelline space, if this mechanism fails, it will lead to polyspermic fertilization, which will lead to formation of polyploid embryos. Normally the human body or the human body cells, they are in diploid state, and this zygote as well, because female pronuclei is single N, which is haploid, and the male pronuclei is also in haploid state. Once these two combine, the zygote will be in diploid state. But if polyspermic reaction or a polyspermic fertilization occurs, there will be more number of these male pronuclei, which will lead to formation of polyploid embryos, which obviously won't be able to survive and will undergo embryonic death or lead to abnormal development, and later on monsters will be produced instead of this normal fetus. So this is a pictorial depiction that how these things occur. See, the sperm gains entry into the perivitelline space. It enters the this inside the sperm pronucleus, enters into the female nucleus, this egg nucleus, sorry, and the second polar body is released. See, these are two polar bodies. Earlier, there was just one polar body, and once this sperm head gains entry into the perivitelline space and fuses with the zona pellucida, second polar body will also be released. This is male pronuclei and this is female pronuclei. They come close. The nuclear envelopes, it gets dispersed or disappeared, and these chromosomes, they combine together to form the zygote. So this is another picture showing this fertilization. So this is the picture that this is the picture of under stereo zoom microscope. So once this zygote is formed, it will appear like this. This is first polar body or second polar body. This is another polar body, and this is male and female pronuclei inside, which are clearly visible. So this is the normal procedure, this is the normal process of fertilization. So there are few factors which may hamper this fertilization process or which will lead to fertilization failure, even if successful mating occurs. So these causes can be if there is any abnormality with the ovulation process, or there is sperm and its transport abnormalities. Either the sperms are defective or there is some aberration in the transportation of the sperm cells through the cervix, through the uterus, through the oviduct and into the oviduct. Or there is some endocrine imbalance, because as I told you earlier also, that this balance of hormones is very important for successful transport of this sperms into the female genitalia, as well as proper ovulation to occur, or the transport of the ovum through the oviduct towards the uterus. So all these events, they are very well these controlled by these hormones. So if there is any imbalance of hormones, either any of these processes will be hampered, or all the processes can be hampered, which will lead to failure of fertilization and early embryonic death due to any infectious reason, or due to this implantation failure, or any of the reasons. If early embryonic death occurs, so it will also be considered as failure of fertilization. Though fertilization was there, but there won't be any successful pregnancy, right? So all these events or all these causes, they can lead to failure of fertilization. So that's all for today's lecture. Thank you very much. If there is any confusion or query, you can ask, because we still have 5.5 minutes remaining for this session.